You're going.
Hello. Welcome back to PMB 150 Final Review. Today we're talking about literally three things. If you wanted to learn about electron microscopy, you should have been here because Allison did that already. But I will tell you about mitochondria. This is the lecture I don't feel incredible about, so I'm going to just tell you things, which is that mitophagy happens, and it happens a lot because mitochondria be having reactive oxygen species. So we got to be engulfing. So mitophagy meaning like take it apart. We need to recycle you. This mitochondrion is bad. But I don't really know how that works. But anyway, when it is decided that this shall happen, there is a garbage tag that is bestowed upon the mitochondrion of target, and that is called TRAB. That's an outer membrane protein, obviously, outer mitochondrial membrane protein. Because if you're going to be it's a tag, right? To be detected by something else, it needs to be on the outside. Because how is it going to find you if you're already inside? So it'll bind to VAP27, this transmembrane protein on the ER membrane. And so imagine this, except we don't actually know if this is exactly correct. But what will happen when we talk about autophagosome formation is they come from the ER membrane. So we kind of feel like this might be a situation that happens, is that we got like invagination budding off. I don't know. We don't. Maybe it doesn't work exactly like that. But anyway, it binds to this, and then next step is the phagophore development, and we have our mitophagy. And this happens through ATG 5712. Choose to memorize that or don't, is up to you. What are some functions of the mitochondria? Moving on. We got energy, obviously the powerhouse of the cell. And that's kind of related to redox. You know, we're going to be making reactive oxygen species as we do our TCA. TCA? Yeah, that happens there. Electron transport chain things happening. Detox, biosynthesis, those are all related. We also have calcium signaling. Somehow depends on inner mitochondrial membrane potential. How? Also programmed cell death, which is related to like autoimmunity, the hypersensitive response, but also normal cellular activity such as like leaf spins. We also see fission and fusion of mitochondria. This is still something that we don't really understand super well, but it's very interesting. So obviously, fission, like if you want one more mitochondrion, you need to make another. And the way that it does that is through fission. So there is, when we want to make two, the site that it will divide at is determined by contact with the ER. And then you'll have some familiar proteins that we talked about yesterday. Fis1AB, which works with another protein, PMD1, which recruits L1, which recruits DRP through AB. So these guys are familiar. So these are dynamin-resembling proteins. Dynamin, remember, helps our CCBs bud off. And so similarly, we're kind of like condensing, and this is the same thing that it does for peroxisomes, and then that results in fission. And also we have the accumulation on like this fission site in the membrane of cardiolipin, thought to play a role in facilitating that. Anyway, if we want fusion, you'd say, okay, if we're fissioning, making more mitochondria, why would you want to fuse? Still kind of unclear, but definitely what's true in plants is that on average, there's less than one mitochondrial genome per mitochondrion. So that can be a problem. So you want to have one genome per mitochondrion, you're going to want to fuse with somebody else. And so what happens is you have these, on your outer membrane, you'll have these GTPases that can dimerize. And so that tethers your two mitochondria together. And that's myro 1 and 2. And then as a GTPase, they hydrolyze GTP, which helps the membranes to fuse. And then also PMF, promoter mitochondrial fusion, is involved. Moving on. The autophagosome. Let's eat some stuff. Especially when we're in nutrient deprivation, because that's when you gotta eat, is when you don't have a lot of nutrients. You gotta recycle some things that might not be as important. Remember I said it comes from the ER, and you have this general cycle. There's some things going on here, and we'll talk about that in a sec. But basically, you have the ER, you bud off your phagophore, and then you will have vesicles delivered to you to expand, and as you wrap around your target thing, and then you will be a full autophagosome and go to the lytic vacuole. The thing that you will probably engulf is likely something with a lot of ROS. So chloroplasts, mitochondria, peroxisomes. Make sense? We'll get to the numbers in a second. But yeah, no, that's how I feel about that. It's double membrane bound. Makes sense? Of not that many things in the cell. We got our plastids, we got our nucleus, we got our autophagosome. So let's start at number 1. How do we get a phagophore budding off of the ER? Okay, so our nutrient deprivation guy, SNRK1, will phosphorylate this guy, Raptor. This is a subunit of TOR. That's a lot of things. So when Raptor is phosphorylated, it will stop TOR from doing what it does, which Raptor is a part of TOR, but it'll be like, hey, stop doing what you do. TOR is a kinase, so obviously it's going to be phosphorylating things. And what it does is it phosphorylates this guy ATG13. When ATG13 has been phosphorylated, it can't do the thing that it normally does, which is bind to ATG1, which has been phosphorylated by SNRK1, who, remember, they be phosphorylating things for nutrient deprivation response. Involved another nutrient signaling. But that's not relevant. I mean, it is, but not in this class. And then we have ATG1 and ATG13. They have been bound. Technically, ATG13 that was phosphorylated. Oh wait, no, ATG1 was phosphorylated.
No, 13. No, wait. Yeah, 1. I'm wrong. Sorry.
Well, I did two wrong the first time. And then we have this results eventually downstream in phagophore formation. So that's step 1. We have the phagophore. But remember how I said we have vesicles being delivered to it to, make more? Well, you want to make sure, first of all, that the vesicles are being delivered here and not like here, because that's not going to result in that shape. So that's what I draw here. Like, we don't want our vesicles to be delivered on the side. We want it to be where it's growing. And so we have markers called PI3P, which is added by the PI3K complex. And that tells ATG9 to 18 to add vesicles where PI3P is. And then you get your expanding phagophore that will wrap around your target. But how does it know if it's wrapping around the target? Well, it has some interior, like on the inside of the forming autophagosome, membrane proteins that will bind to receptors on its target. There will sometimes be some on the outside. We think those might bind to microtubules for like cellular trafficking. This is a little unclear. But anyway, these guys are called ATG8. And just a plain old ATG is not ready to be doing its job. First, it needs to have some post-translational modifications. So the C-terminal glycine will be removed, and instead will be added PE, it's a phospholipid, which makes sense because if it's in the membrane, it's got to have a phospholipid. And so yes. Mitochondria, nope. Autophagosomes, well, we did talk about those. Anyway, final step. Membraneless compartments, also known as biomolecular condensates. This is my best recreation of it. This is our cell. There are other things not pictured in the cell. Why? Basically, it's like a bunch of stuff. So like a clump of things. So we can use it to sequester unwanted proteins, or we can use it to like release them in a time-specific manner. So like temperature, if you have a bunch of things that are wanting to be released at a certain time, like maybe you detect temperature and you're like, it's time to flower now. You might have ELF3 being released in a condensate that results in early flowering 3. So how do you figure out what is in there? What in there? You can do that. You can figure that out through liquid phase separation. Oh, it's too lazy to write liquid twice. And through some process. This is the question mark. It's like we know what it is, but it varies per biomolecular condensate that you're looking at. But basically, you do something so that the solute interaction, so it interacting with itself, are more favorable than the solute-solvent interaction. So that will result in it like diffusing. That does not tell you what it is. So you do something. So in the example of like your FG noobs, remember they're very gel-like, kind of hard to like condense and extract, I guess. So if you do some editing and you change the phenylalanine of the FG to serine, you can then dissolve that in water, which is interesting. Important to know in your LLPS is if it has to be for something that like has multiple surfaces. Because if you have like something that can only form a dimer, like this, it only has two pieces it can only interact on two faces, then like you can't make a big clump of it because there's not going to be another side for you to add to. So in here we were like interacting on all sides. It's a multivalent interaction. Pretty cool. And this can be a protein surface or an intrinsically disordered region, or maybe both. I don't know. I mean, I don't see why not. Also, they don't move with cytoskeleton. Fun fact. I don't want to talk about this. Okay, good luck, team.